Development of dicot embryo. The first, that is the, suppose this is the dicot embryo, that is diploid zygote. One is the chalazal end, other one is the micropyle end. And in before after fertilization we have seen this polar nuclei, this PEN will develop. Fertilization will take place in double fertilization. One male gamete will fuse with two polar nuclei, that will develop into PEN, primary endosperm nucleus. From this primary endosperm nucleus, primary endosperm cell will develop. From here the endosperm will develop, and endosperm will be going to give the nutrition to the growing embryo. So now the embryo, how it divides, that we first learn. So first there is a this is a embryo, dicot embryo, and suppose it's a dicot embryo, 2n zygote, the diploid number of zygote is there. This is the chalazal end, this is the micropyle end. Now its mitotic division takes place, and this zygote divides transversally. Okay, the chalazal end part will give rise to terminal cell, and the micropyle end part will give rise to the basal cell. Now this chalazal cell and so now this terminal cell and the basal cell will give rise to different shape. The terminal cells will again divide into four cells, and they are known as proembryo. So terminal cell divides to form proembryo, and the basal cell will divide transversely. The basal cell undergo transverse division. So first one basal cell will divide into two, then again this one divide into two. In this way the division will take place, and the last one cell that is in the large in structure, that is known as hostorial cell. And these transverse cell that develop from the division of basal cell is known as the suspensor cell. So what is the role of suspensor cell? They are pushing the embryo deep inside the endosperm, that we will come later, this function. So after this what will happen? After this I draw the structure here. You see this proembryo, we call it quadrant, proembryo quadrant. In some books it is written quadrant because it has 4 cells. Now this quadrant will again divide into proembryonal octant, because you see 8 cells are there. Clear? Acha. Now the cell which is now the suspensor cell which is attached to this proembryo cell, it is now big enough. You see it's now big enough. I have drawn it with different color, and it will call hypophysis, and the lower cells are all suspensor cells like this. The first suspensor cell which is attached to the proembryo will give rise to hypophysis. Now the lower cells are suspensor cell, and this is the hostorial cell. Clear? Now what will be the next change? In the proembryo octant we say this is proembryo quadrant because 4 cells are there. It is proembryonal octant because 8 cells are there. This proembryonal octant again divide into inner and outer cell. You see, and this in this way, this cell will divide like this cell will divide like this, in this way. So division takes place, outer inner, outer inner, outer inner. So 16 cell we will see here, and the total structure is known as dermatogen, which will give rise to epidermis later. Okay, this is the part of epidermis. Okay, dermatogen cells. And the hypophysis again will divide hypophysis. This hypophysis again will divide, and it will give rise to 1, 2, 3, 4, 5, 6, 7, 8 cells. And below the suspensor cell and they are there, and the hostorial cell are exactly like this. So you see the hypophysis divides and form 8 cells transversely. The lower 4 cells will give rise to root cap, because from these the seed will germinate. Okay, so lower from here, the lower 4 cell of hypophysis will give rise to root cap and the root tip. The upper 4 cells of hypophysis will give rise to root cortex, and the 16 cells, out of which 8 cell which is with the touch of this hypophysis will give rise to hypocortile. What is hypocortile? I am telling you later. And the upper 8 cells which are not in the contact with this hypophysis will give rise to plumule and cotyledon. So basically you remember, from plumule the shoot is developed in a seed. From plumule the shoot is developed, and from radicle the root is developed. Okay, radicle means this root cap, root tip and root cortex together called the radicle. Okay, so this is the thing that happened. So in a anatrophous ovule we have seen that already the after double fertilization the endosperm already start forming, and this embryo, we know that now the dicot embryo will also start. So this, as I written here, the suspensor cell pushing the embryo in the endosperm. Why this suspensor cell pushing the embryo inside? To get the nutrition, because endosperm will give it the nutrition. Clear? So this is the endosperm, and this suspensor cell pushing this upper part towards the endosperm, because we know endosperm will give rise to will give the nutrition. So now what will happen after this? After this what will happen? So from we have seen this structure. So this portion, the cell of this portion means this dermatogene cells. From here, as I told you, plumule, cotyledon and hypocotyle will form. So you see, this cell will again divide and it will form a mass of cell, and here these are the two cotyledon will develop. Between two cotyledon there is a structure that is known as the plumule. From plumule the shoot will develop, and these are the two cotyledons, and the lower hypophysis cell and below the suspensor cells are there which will degenerate, and this hypophysis cell which have divided into 8 cells. As I told you here, that the upper 4 cells will give rise to root cortex, and the lower 4 cells will give rise to root cap and the root tip. So from here this cells, upper cells will again divide like this, and the lower cell will again divide, and it will form a mass of cell, and the lower cells which will give rise to the root cap and the root tip that are known as radicle. As I told you, this together called radicle, and the root cortex that will give rise to hypocotyle. Now you can ask me, ma'am, what is hypocotyle? I can't understand the shape. Okay, now I am telling you what is hypocotyle, what is epicotyle. I am telling you later. But this structure of seed that is known, dicot seed, is known as globular structure. So from this globular structure, what will generate the heart-shaped structure of the seed? You see, this is the heart-shaped structure of seed. Okay, because you see the two cotyledons are very close to each other, they will not stay in this form. So these cotyledon will come like this, and these cotyledon will come like this. They will bend towards one side, and that structure is similar like the horseshoe shape. So first, from the globular shape, the seed will change into heart-shaped cell. From the heart-shaped cell, the seed will change into horseshoe-shaped cell. So now what will we see? The actual size, seed of a dicot plant. Here I have drawn the structure. You see, this is this proportion is the two cotyledon. Okay, this is the embryo main axis. Okay, so here you see the cotyledonary node. The two cotyledon are joined with this axis with a cotyledonary node, and the upper part of this axis that is called as plumule, and the lower part is the radicle. So radicle, from here the root will develop, and plumule, from here the shoot will develop. And cotyledon, after endosperm, endosperm will store the nutrient in the cotyledon. Okay, so when the endosperm totally used in this place, now when the seed will form this structure, the endosperm will totally used, and some part of the endosperm they deposit their some nutrition in the cotyledonary part. Okay, so when from this seed the new plant will germinate, they use the nutrition from these two cotyledon. Okay, so when from a seed, when a new plant will develop, the two cotyledon will these two cotyledon will disappear at this time, that we know from the lower classes, right? Acha. Acha, the space between the pimule and this quaternary node is known as epicotyl, as I told you. Is epicotyl and hypocotyl, as I told you na here. Hypocotyl, and this is the epicotyl. Hypocotyl is the lower part. The space between the quaternary node and the radical is the hypocotyl, and the space between the pimule and the quaternary node is the epicotyl. Okay, so this is the structure. Now we have understand from which part this structure is generating. So in this way the dicot seed generate.